afternoon. Thank you for joining us for Research 411, a talk show focused on sharing best practices and trending research topics delivered by professional staff and prominent UT Dallas researchers from various disciplines. I am Tiffany Willoughby, Assistant Director in the Office of Research and Innovation. I will moderate today's discussion. During today's talk show, Dr. Gagan Wig will discuss his Alzheimer's disease research and how lifestyle and environmental factors play a role in the disease's progression. Dr. Wig is an associate professor in the Center for Vital Longevity and School of Behavioral and Brain Sciences at the University of Texas at Dallas, and in the Department of Psychiatry at the University of Texas Southwestern Medical Center. Dr. Wig earned his bachelor's degree from the University of British Columbia and his Ph.D. from Dartmouth College, followed by a postdoctoral fellowship at Harvard University and Washington University in St. Louis with the Human Connectome Project. Dr. Wig's research uses functional and structural imaging to measure the organization of human brain networks, specifically focused on understanding how brain networks change across the adult lifespan in both health and disease. Dr. Wig's recent work is determining how changes in an individual's socioeconomic environment and lifestyle interact with their brain network organization to predict their resilience and vulnerability to cognitive decline. Dr. Wig's research is supported by grants from the National Institute of Aging and the James S. McDonnell Foundation. Let's jump right in, Dr. Wig. Can you start off by explaining what Alzheimer's disease is and telling us about your research at UT Dallas?
Hi, Tiffany. Sure. Alzheimer's disease is a disease of aging. It's the most common form of dementia, and that's characterized by initially by memory loss, and then eventually more broad impairments in executive function and even sensory processing. I think many of us when we think of Alzheimer's disease, at least biologically, we think of some of the characteristic pathologies that are associated with it, which are amyloid plaques and neurofibrillary tangles. Our research is trying to understand why people are more vulnerable to Alzheimer's disease. We think about aging across the healthy lifespan, but also in context of disease, and Alzheimer's disease is one of those diseases we're interested in better understanding.
Thank you very much, Dr. Wig. Now, tell us a little bit about how the brain works, how the different parts of the brain actually work with each other.
Yeah, so, I think we've known for a long time that the brain, the way cognition works in the brain, isn't via the recruitment of single areas that are dedicated to, functions like memory, right? So one area won't map onto things like memory or language, but rather those functions are a product of distributed processing across multiple areas. And an area is a collection of neurons. So what you essentially have is functionally distinct areas of the brain, and they all interact to give rise to your ability to remember your past, to attend to your cup of coffee and reach out and grab it, and so forth. So the way we've been approaching healthy aging is by thinking about those network interactions, and they really are embedded in a network, much like a social network or a technological network. So we think about how areas interact with one another to allow you to do these myriad of things.
Okay, and do you have a certain tool or network tools that you use?
So we use non-invasive brain imaging, so functional MRI. Where we're trying to, where we measure correlates of blood flow. So when neurons are active, when doing something, they require energy, and that energy is delivered by blood flow. So using an MRI scanner, you could measure properties of that blood flow as things change and as activity changes. So we use MRI scans and we measure functional correlations across regions. What that means is we're looking at how different areas of the brain, when someone's awake and lying in the scanner, are correlated or are co-activated, even in the absence of doing anything.
Very interesting. Now, what prompted you to start researching or what inspired you to go into researching Alzheimer's disease?
Yeah, I mean, that's a great question. We've been really focused on thinking about healthy aging. Even in the absence of disease, cognition changes from birth up until old age. Sometimes not for the better. So there's variation and there's change, but on the extremes, there's illness. So I studied I studied in systems memory, and got interested in thinking about aging and the problems of aging and how aging is associated with memory loss, even in kind of in the normal range. But obviously, the more extreme versions of that would be disease of the brain, which most prominently is dementias, and Alzheimer's disease is, as I said, the most common form of dementia.
Now, can you tell the audience what causes Alzheimer's disease specifically?
Yeah, I mean, this is the million-dollar question. So there are no known determinants of Alzheimer's disease. There are risk factors that have been linked to increased risk. And in some, there's rarer forms of Alzheimer's disease where there are dominantly inherited genes that would result in Alzheimer's. But by and large, the majority of cases of Alzheimer's disease are of a different form, which is called sporadic or late onset. And there are genetic variants that result in increased risk, but as I said, there are no determinants. So that leads us to understand that there are other factors at play. And one of the big factors at play are environmental moderators, right? So we know that two individuals with comparable genetic risk of Alzheimer's can end up on different trajectories and different endpoints, right? One might succumb to the disease and another might not. So then the question is, what are the health, lifestyle, and environmental factors that might be, leading one person to the disease state and the other to be relatively healthier? And so that's what we're trying to understand. What are those exposures that result in disease, and which are the exposures that might protect you from disease?
Very interesting. Now, can you tell us a little bit about why the risk of Alzheimer's disease increases as someone ages?
Yeah, I mean, so, ultimately, there's increased prevalence with increased age, right? So, starting from about 65, you start to see at least the prevalence increase in the population. For the individuals 80 and above, there's like 50% prevalence. So it's clearly linked to the aging process. The pathologies increase as we grow older, and ultimately it's caused by neuronal loss, so loss of the cells of your brain. And there's various ideas about why that might be the case and why aging, that as we age, those processes occur with increased frequency. But there's still a lot of unknowns.
That makes sense, and it makes me curious to know whether or not, environmental influences coupled with the aging brain also makes the risk higher for an older individual to develop Alzheimer's disease. Can you weigh in on that?
Yeah, certainly. I mean, that's exactly the thinking. As we grow older, our exposures accumulate. So, in the case of health behaviors, negative behaviors, or absence of adequate exercise or adequate diets, those things will accumulate and continue to wear and tear on the body, but also the brain. So, the exposures do seem to matter, and we're trying to understand what those exposures are that are most going to lead you to be most vulnerable. And we're also trying to understand better whether or not we can identify those exposures that are presumably modifiable early on. I think that's one of the key things in Alzheimer's research, right? We're seeing a lot of reports now about the ineffectiveness of certain drugs. But one caveat to a lot of these studies is that that work is done typically on individuals who might be already impaired. So, much like other diseases of the body, a prevalent idea is that what should be done is trying to detect the onset of pathological processes earlier on, and that would give us a better opportunity to intervene, even via modifying lifestyle, right, or making recommendations.
Can you address some of those first signs of Alzheimer's disease? What are some of the symptoms?
So, in terms of behavior, the classic symptom initially is a report of memory impairment, and that comes from either self-report of subjective memory complaints or a loved one indicating that memory is failing. And I guess it's important to point out that as we grow older, memory declines in general. So there's normal variation, but really what Alzheimer's is characterized by is more extreme degradation. It's normal for us to all, lose our memory to some extent, right? My memory isn't as strong as it was 15 years ago, but in terms of the disease, it's these sudden and abrupt and really, kind of noticeable impairments that are quickly changing.
Very good, thank you. Now, I know that you previously spoke about brain networks and mapping. Where do you see the future of this mapping going? And will I ever be able to go to the doctor and get a test to find biomarkers that will help me to prevent Alzheimer's disease?
Yeah, I mean, this is exactly what our goal is at this point, right? So I mentioned the fact that, often it seems like characterization of Alzheimer's is already associated with frank pathologies. So the goal for us is to identify biomarkers or signals that are detectable and that we can measure early on. And in my lab, we're really focused on thinking about the network organization of the brain and how it might change. We think it has information, even in the absence of pathology. So I have a slide that we can share with the audience, just to give people a deeper sense of what we try to do. So as I said, we use functional scans or MRI scans to measure brain network organization. On the left is just a representation of areas of the brain depicted on an inflated cortical surface of the left hemisphere, kind of looking at it from the side. And what we can see on the right is this is a graph representation of two individuals and how their brain networks are actually changing. Now, each of those colored dots is actually an area of the brain. And the position of those dots isn't based on the location in the brain, but rather it's based on how strongly it's associated with other dots. So you could think of these like friendship networks, right? If you kind of created a network of all your interactions and all your relationships and connected people via these links and dots, people that had stronger ties or affiliations might be closer to one another, and groups, you would see groups of individuals and so forth. And that's really how we approach the brain, right? We're thinking of areas of the brain, how they interact with one another, and how they form groups or communities with other areas to allow us to do things like see the world or remember our past. And it doesn't take a network scientist or a neuroscientist to appreciate that these two individuals seem to be on different trajectories, right? The individual on the left, their networks are pretty maintained across 6 years even. The extent of their brain network changes are not very large. Whereas the person on the right, something's happening. There's a greater clumpiness, and we have measures to characterize that clumpiness or the kind of what's called desegregation of the network. And we've found that it actually is linked to worse memory function. It's associated with various demographic variables linked to SES, socioeconomic status, and it's also those kinds of patterns are prognostic of clinical impairment or dementia severity. So that's essentially one way we're thinking about measuring brain health, right? I think a lot of what people are familiar with is thinking about the structure of the brain, the size of different parts of your brain, or even characterization of pathology. And we've really been interested in this other angle, which is thinking about how brain areas interact with one another and how those networks might be breaking down. I think there's a lot to learn from this approach.
That's very interesting, Dr. Wig. Thank you for sharing. Now, once someone is diagnosed with Alzheimer's disease, can you slow or reverse the progression? And if so, how do you do that?
Man, that's such a critical question, and one we all want, we all hope there to be a more definitive answer to. At this point, there's ways of alleviating some of the symptoms. There's some pharmacological agents that work on acetylcholine, which some physicians might prescribe in some cases. But by and large, we don't currently have any treatments for either slowing or halting or reversing the disease. Of course, this is a major effort from researchers and other companies, but that's the goal, is to understand that. And our angle on a lot of this, as I said, is to think about whether or not we can detect things earlier on, because I think there's better opportunities there, much the way that there are, we see with cardiovascular health. At present, you go to your physician for an annual visit, and they'll measure your blood to characterize cholesterol levels and other measures of cardiovascular health. And they could make recommendations to you. We think, you need to exercise more, eat better, and so forth. And I think we take that seriously, right? When we're told that we're on a path, a negative trajectory, we, at least in the short term, make those modifications, which I think has been critical in addition to innovations in other ways of alleviating cardiovascular disease. But we don't really have that for brain health, do we? At this point, there's not much that is measured or detected or done. And that's the goal. If we can start getting those measures identified and administer them and quantify them at an early enough time, then we might be able to say, okay, you're on a trajectory where there's risk of negative outcome. Perhaps we can use these modifiable interventions, and the interventions might not be very different from what we're used to. Eating better, exercising more, but maybe even things more geared towards the brain, being told to engage more with new learning circumstances and so forth. But I think at least in that situation, rather than just being told these things are good, you have some definitive reason to try to change your lifestyle for the better, so that you can, and then get some feedback, right? Is whatever I'm doing resulting in improvement in these risk markers? I think that's the goal for a lot of this.
Perfect. So early detection is best, and then hopefully implementing some of those modifiable factors, exercising, eating nutritious, sleeping well, and maintaining a healthy relationship will help individuals down the line. Thank you. Now, how are you going to isolate factors that contribute to Alzheimer's disease and age-related decline more broadly? It seems complicated to differentiate age from environment and other factors.
Yeah, that's a wonderful question. We're really trying to understand exposures really broadly. I think, a lot of the things you just mentioned are things that we can measure, with mixed effectiveness. Some require self-report, others can be done with biosensors, others can be done with blood work, right? Or at least outcomes can be quantified with blood work. But we are really trying to understand the exposures of an individual, both in terms of their daily living, how much they're sleeping, how much they're exercising or moving around more generally, the nature of their diets, both either via direct questionnaires, but also via measuring cardiovascular health and measuring cholesterol, but even more broadly thinking about stress levels. Stress comes in lots of forms, different magnitudes, different types. We think stress plays a large role in the aging process. Ultimately, you're right, there might be some divergence, right? The things that result in accelerated aging might be distinct from the things that result in Alzheimer's disease or the instantiation of Alzheimer's disease. But the goal right now is to try to establish ways of quantifying these difficult to measure exposures, and then also understand how those exposures changed over the life course.
Thank you, Dr. Wig. And can you tell us a little bit about your new study?
Yeah, so the new study is trying to do exactly just this, right? We recently have demonstrated that those brain network measures change as we grow older, and they're linked to distinctions in demographics and measures of socioeconomic status. They're also prognostic of dementia. So the question now is, can we detect those changes earlier on in adulthood? And we're interested in middle age right now, because I think there's opportunity there to modify lifestyle and habits in a way that might slow down or minimize the impacts of later age brain disease. So the new study we're collecting is based in the DFW area, and we include extensive measurement of brain network organization, but also other measures of brain structure and function. And then we're measuring a whole host of things to try to understand health, lifestyle, exposures. And we do that via some of the methods I mentioned earlier. Much like, the goal is to do this longitudinally, meaning we bring people back over time and see how things change. One broader question we're asking is there something about individuals' brain networks that makes them more resilient to the onslaught of negative exposures? As I mentioned, two individuals might start out with similar genetic risk, maybe even comparable cognitive profiles, but they can end up in very different places. And we think that certainly is a product of something happening in the brain and interacting with exposures. And we're trying to understand whether or not we can identify that combination of things that makes you more or less vulnerable.
Are you actively recruiting people to participate in this study?
We actually are. We are actively recruiting. We can share a link with you or with the community if they're interested in seeing if they're eligible to participate. Yeah.
Excellent. Thank you, Dr. Wig. And I believe that we have a few questions in the Q&A chat from the audience, and I'll go ahead and deliver those questions to you. Is the difference between normal aging and Alzheimer's only in the degree of loss?
Oh, that's a great question. No, so as I mentioned, the hallmark of Alzheimer's are the presence of at least two forms of pathology, beta amyloid plaques and neurofibrillary tangles, which are tau tangles. And while we can see that in the absence of those pathologies, in the absence of disease, one idea is that maybe that's a pre-symptomatic version of the disease, right? But aging and Alzheimer's disease are distinct in that way. Healthy aging is defined by the absence of pathology.
Thank you, Dr. Wig. The next question in the queue is, how do researchers try to weigh or differentiate the various environmental exposures that may slightly or severely influence the likelihood of developing Alzheimer's disease?
Yeah, wonderful question. I mean, what a challenge, right? To try to understand this complex myriad of exposures that are impacting our health and our cognitive function. So there's various approaches. One is you can think of the epidemiological approach, right? So characterization of habits and exposures in large cohorts of individuals and looking at their trajectories. So it's observation-based studies. You could also do that on a smaller scale where you've targeted maybe certain types of environmental exposures and you try to measure them with more directed techniques, as I alluded to earlier, biosensors to measure activity levels or sleep habits and so forth. But then, of course, there's these would all be in human studies, and there's also a whole suite of tools that we have in animal work, which do provide us insight, right? So studies of mouse models of aging, for example, where there's been manipulations of exposures and manipulation of adverse events at various stages of the lifespan. Those two can give us a deeper understanding of mechanism. And then the hope then, of course, is that we translate that upwards to humans by understanding if those are also present in the observational work.
Thank you, Dr.
Oh,
Wig.
and then I guess the final one, sorry.
Sure.
And then there's interventions, right? So there are certainly interventions where there's an idea of something that matters, and then you actually do ideally a double-blind control study, and have one group do the intervention that's hypothesized to matter, and another group do be in a suitable control condition, and see whether or not the metric of function or health actually changes.
Thank you for answering that audience question. All good information to share for those interested in conducting research. Now, next question in the queue is, for Alzheimer's disease, usually the pathological changes happen in the cellular level, like the aggregation of the amyloid and tau protein in neuronal level. How the network theory, graph theory in the brain regional level captures the early change in cellular levels. It looks like a word was left out. So how do the network theory and graph theory in the brain regional level capture the early changes in the cellular level. Can you address that, please?
Yeah, so how do we link those scales? So, even the pathology occurs at the cellular level, but it occurs in a way that accumulates in regions, right? So, one of the earlier regions of devastation in Alzheimer's is the hippocampus, right? And then there's characterizable patterns that evolve. So while they happen at the neuronal level initially, those things aggregate, right? And ultimately, Alzheimer's disease and these other dementias are a consequence of degeneration, of neuronal death. And that isn't one neuron dying, but many neurons dying. And given they happen in these groups, those groups are actually, those parallel the groups of regions that we look at in terms of network organization. So while our scale of analysis is a bit broader, we're thinking about the cities as opposed to the individuals. The patterns that happen in individuals amalgamate, and we can see those signals change at the city level or in the area level of areas of the brain. So that's why the network-based approach, which is amenable to neuronal connectivity as well, but we're able to do it at a more broader systems level of organization. Yeah, good question.
Thank you, Dr. Wig. Another question that we have, kind of related to some conversation we had earlier during the talk show, do you have any additional healthy practices to suggest that individuals can put into play in their lives to decrease the likelihood of developing Alzheimer's disease?
Yeah, wonderful question. I mean, I wish I had, the magic answer at this point. All the evidence seems to suggest that, one way of thinking about it is what's good for the body is good for the brain. So a lot of the things that we're, told to do to maintain and promote health of other systems seem to be beneficial to the brain, as we've been mentioning a few times over, exercise and having a healthy diet. But other factors that seem to matter are getting adequate sleep. And in addition, lifelong learning seems to really matter, right? So this idea of maintaining cognitive activity, right, and cognitive vitality seems to matter. Now, this doesn't mean you should jump online and play a bunch of video games. I think we're still trying to pick apart, what the nature of that cognitive activity is. That this seems to matter. But I think there's good evidence to believe that if you continue to learn in an engaged way, that does seem to help at least maintain cognitive health. And one of my colleagues at CVL, Denise Park, has done a lot of work on this area, and she's guest on the show previously, I think.
Yes, she sure was. Thank you. What are preliminary findings? How long has the study been going on? And they're referring to your study. And how long do you plan for the study to continue? So this is the longitudinal study, I believe you referenced.
Right. So we don't have any preliminary findings yet. I mean, the study is built on a number of papers that we recently published. Most recently in November, there was a big finding that we made that linked those network changes to prognosis of dementia, as I mentioned. But as yet, we're actively enrolling participants. And we don't peek at the data until we've collected all the data. But it's, as I said, it's a longitudinal study that's supported by the NIA, National Institute of Aging, and we're planning to have it carry on for at least 4 to 5 years.
Right. Okay, looks like we have 2 or 3 more questions in the queue from the audience. Do you think, Dr. Wig, this period of COVID has changed anything related to the onset of Alzheimer's disease?
I do. I mean, we're in this unfortunate circumstance where many of the things that I've mentioned are much more difficult to engage in, right? So, I mean, this is a time where it's been challenging to exercise in a safe way. It's been challenging to meet with our friends and loved ones and socialize and maintain, visit museums or just even go out and chat with friends and spend time. And of course, there's been a lot of sleep loss, and there's been increased stress from a myriad of things that are associated with the pandemic. And unfortunately, that's the outcomes there are not a great one, right? There is data now indicating that a diagnosis of Alzheimer's disease has increased during the pandemic.
Thank you for sharing that information. The next question refers to the Nun Study. The Nun Study was an environmental study, but it showed some nuns that showed signs of Alzheimer's, but after death, there were no signs, and vice versa. Is there new data on that? If you have studied it or have information to share, we would love to hear your feedback.
Yeah, good question. So, it wasn't the case that, so at post-mortem, there was evidence that some of the nuns did exhibit some of the pathologies that are used to define Alzheimer's. I think the really interesting thing about that study is the observation of the extent of the variability, right? So these are a group of individuals that, are living in their similar circumstances, but also probably have a variation in their exposures, right, and the types of things they're engaging in. It wasn't the case that there was a one-size-fit-all in terms of their cognitive health or their cognitive trajectories. Some of those nuns did stay cognitively healthy, and others not so much. So there's been a lot of work on trying to understand, again, whether there are certain brain-based markers that would have allowed researchers to predict who would have stayed healthy and who wouldn't have, in addition to understanding whether or not the exposures of certain nuns were predictive of the outcomes.
Thank you, Dr. Wig. And last question today from the audience refers again to your longitudinal study. In such longitudinal studies, how often do we measure to analyze the pattern of change?
Yeah, great question. I mean, this varies depending on the goal of the study and the feasibility. You can measure, for our study specifically, there are things that we're measuring daily, and there's other things that we're measuring over multiple years. So, in terms of one of the primary goals, though, is to understand brain and cognitive changes. And those changes, while they vary, the cognitive changes could vary, month to month, and so forth. The brain changes, we believe, to really kind of take hold over a period of a few years. So our goal is to measure every 2 years in this longitudinal study, so that we can detect some of the bigger changes of brain network organization and cognitive function.
Well, thank you for joining us today, Dr. Wig, and audience, thank you for attending our program. If you enjoyed today's talk show, please suggest it to a friend. To stay abreast of Office of Research and Innovation information and programming, please follow us on Instagram and Twitter and subscribe to our newsletter. Feel free to use your phone to scan the QR codes on the slide or follow the links posted in the chat. We look forward to having you join us for an upcoming Office of Research and Innovation event. Thank you and have a nice afternoon.